Biotechnology Part 1, Yeast and Pharmaceutical Industries. So if we have a look at yeast industries, first of all. Firstly, we look at types of microbe. There are four main types: fungi, of course, like mushrooms, toadstools, and yeast; bacteria, viruses, and single-celled animals or protozoa. So if we have a look at microbes in industry, bacteria are used to make cheese and yogurt, and yeast, of course, is used to make bread, wine, and beer. In bread making, the reason why we use the yeast, it makes carbon dioxide gas, which enables the bread to rise. In making alcohol, by a process of fermentation, sugar is broken down on its own without oxygen into alcohol, carbon dioxide, and a little bit of energy. Beers and alcohol in general can be made in batches by batch processing. In terms of beer, you can have two basic types: cask conditioned beer. The yeast remains in the cask and sugar is added, and you get a nice rich dark ale produced. Brewery conditioned beers are your pale lagers, and in those cases the yeast is filtered out. Now, different types of alcoholic drink depends on the source of the sugar. Apples are used to make cider, potatoes are used to make vodka, and of course grapes are used to make different types of wine. Distillation is used to increase the strength of alcohol in drinks like vodka and whiskey. The fermentation process itself stops at around about 12 to 14% alcohol, because the alcohol actually poisons the yeast and kills it. Distillation is a way of concentrating the strength of alcohol in the drinks. Kefir is a fermented milk drink in hot countries, and especially at times before refrigerators were used. This is a way of helping to preserve the milk. If we have a look at immobilisation now, this is a process of taking jelly beans and impregnating them with cells or enzymes to carry out a particular function. The good thing about this is that they can be reused, so there's fewer wastes and so reduced cost. But this is part of what's called continuous flow processing, not batch processing. As long as you add substrates into the top of the reaction chamber, as those substrates pass over the jelly beads and come into contact with the cells or enzymes, then the process will continue and you'll get a steady stream of products coming out of the bottom of the reaction chamber. Some other facts about yeast, just to finish off. It can be used to make food flavorings and colorings. For example, red yeast is fed to farmed salmon to make the flesh pink. And waste products from the yeast industries can lead to eutrophication. What happens here? The waste yeast can become a food source for bacteria, and the bacteria proliferate in the water. The bacteria remove the oxygen from the water, and so animal life dies. Yeast can be grown in vast quantities in industrial fermenters, particularly in wine making and beer making. And of course, if you're going to use with microbes, you're going to work with microbes, you need to use aseptic conditions so you don't contaminate the different cultures of microbe you're actually working with. So, moving on to pharmaceutical industries now. Antibiotics. An antibiotic is a substance which inhibits the growth of bacteria, and they are not effective against viruses. Now, if a certain type of bacteria is killed by an antibiotic, we say that bacteria is sensitive to that antibiotic. However, if it's not killed, it is resistant to it. Antibiotics, the reason why they exist in the first place is that they're produced naturally by soil fungi to compete with bacteria. And the first antibiotic was penicillin, of course, developed or discovered in 1928 by Alexander Fleming. Antifungals now are substances which inhibit the growth of fungi. For example, ringworm and athlete's foot. Genetic engineering is taking genetic material from one organism and transferring it into the genetic material of another. For example, we can use bacteria and take little rings of DNA called plasmids out of certain bacteria and insert the human insulin gene. Once we place that plasmid back into a certain type of bacterium, those bacteria multiply and proliferate, and they copy the human insulin gene in each and every one. Then if we grow and cultivate those bacteria in a fermenter, then we can produce vast quantities of human insulin ready to be used by patients suffering from diabetes. So, genetically engineered bacteria can make human insulin, and genetically engineered yeast can be used to make the hepatitis B vaccine. Genetically engineered organisms can be grown on a vast scale, just like yeast, in industrial fermenters, like so. And those growth conditions inside those fermenters can be monitored and controlled by computers. Antibiotics nowadays are generally being overused, and that's leading to antibiotic resistance in bacteria. Now, because of this, we now need a wider range of antibiotics to help treat all the infections caused by these resistant bacteria. Okay, thank you very much.